Hello and welcome students. Today we will be studying about the discovery of cells. So this will be the first lesson of this chapter, and we'll discuss about a world beyond what we can see by our naked eyes. So we can see our arms, we can see our hands, we can see the macro objects very easily with our naked eye. But what are these objects? For example, my skin. What is it made up of? It is made up of cells which we are not able to discern with our naked eyes, because the limitation of the human eye is to observe something. If there are 2 points which are just 0.1 mm apart, the human eye is not able to differentiate between those 2 points. So human eye will not differentiate, it will appear as just 1 point somewhere. Because of this, we need specialized gear to see the cells. So the first time ever somebody who saw a cell was using a cork. So the scientist's name was Robert Hooke. He took a slice of this cork. A cork is basically something that is used to seal bottles. It was used in the older times more often. Now we have our caps. Right now as well, it is used to seal wine bottles and such. So it was first observed by the English scientist Robert Hooke. So he made this compound telescope and observed a slice of this cork, and because of this he observed the dead cells of a dead plant, basically, because the cork is made up of wood which comes from a dead tree or a dead plant. So when he observed, so that he was able to observe the cells. What he discovered in this experiment was that these cells were aligned in a honeycomb like structure. So there was a definite structure that was repeating itself over and over. So if I have to say a honeycomb structure, it would be something like this. So you may have seen a honeycomb and it looks like this. So there were definite shapes that were repeating themselves over and over a period of time. Okay. So he observed that there were various compartments, small little rooms that were repeating themselves over and over. And because of this, he got curious, and also he called these things cells because they looked like tiny little rooms in a monastery. It is where it's written a monastery. You could also refer to them as something like a prison cell. So prison cells are usually same size compartments where prisoners are kept. Okay, so similar to that. And he was only able to see the walls of these dead cells. Eventually, the first look at a living cell came from Antony von Leeuwenhoek. So he used a specialized handcrafted lenses with which he was able to see the living cells within a pond. So he took some pond water and observed it in a slide under the specialized microscope that he had prepared, and he was able to see some small little microorganisms, very small little creatures, and he named them animalcules. So little animals is what that meant. So he could see tiny little animals inside the water that were moving around. At that time he did not know what it was, but now we have identified these as the bacteria and the protozoa. So nearly 170 years after Robert Hooke, there came Robert Brown, who studied plant cells. So he was a botanist and he studied plant cells, and he discovered that there was a prominent dark spot. So there was a very visible dark spot inside the plant cell. He later named this spot the nucleus, which was the controller of cell activity. So it controlled all the activities that the cell did, which was taking in food, taking the nutrition in, excreting that, and all the respiration and all the different activities were taken care by the nucleus. So it was the brains of the operation, and this nucleus also housed the genetic material of the cell. So genetic makeup of the cell was housed by this nucleus. It gave the identity basically to the cell. Now for the formulation of cell theory, Sir Isaac Newton once wrote to Robert Hooke, and he said, if I am able to see further than others, he did not use the word than others, but if I am able to see further, then it is because I stand on the shoulders of giants. So over here he is referring to the people that came before him. So he said that if I'm able to have if I have researched this far, it is because there were people before me who had done some research, and I have taken their research forward, and because of that I have been able to get this far. So basically that is what we see over here. Robert Hooke in his time discovered the cell. Robert Brown discovered the nucleus after that, and then there were various people who came after that, like Schleiden, who discovered that the plants are made up of living cells, and then Schwann, who discovered that all the animals are also made up of living cells. And then there was Virchow, who discovered that the cells are basically how cells reproduced. He discovered how the cells arise from or are made up of previous cells. So they split up and they make new cells. Okay. So this gave birth to a classical cell theory, which stated that all living organisms are made up of one or more cells. So all living organisms, be it plants or animals, are made up of one or more living cells. The cell is a basic unit of structure and function in all living beings. So this is the most basic part of human structure. For example, an organ, we consider humans have organs. Those organs are made up of cells. All cells arise from pre-existing cells through the process of cell division. Any cell that there is, it will be birthed, or it will be born, or it will arise from a previous cell, which will be divided into two parts. Here is a little scale of different things and their sizes. Here is a full human adult. Below that, a smaller scale, you can see ostrich egg, chicken egg, frog eggs, human eggs, and then below that is the size of plant cells and animal cells. After that, smaller are bacteria and mitochondria, flu virus, proteins, lipids, and finally the smallest, atoms. That is what we have learned so far. Okay. So this is I have discussed about this, the limit of resolution. A human eye can see only 2 points as distinct. So human eyes can only see 2 points as different points if they are separated by 0.1 millimeter. So at least they should be separated by at least 0.1 millimeter. Most cells are much smaller than this limit. So most of the cells, the animal cells or the plant cells, are much closer together than 0.1 millimeter. While a bacterium is roughly 1 to 10 micrometer, modern technology allows us to see atoms at 0.1 nanometer. Okay, so a bacterium is roughly 1 to 10 millimeter, a bacterium, and the modern technology allows us to see atoms at 0.1 nanometer, the electron microscope I was talking about earlier. So atoms are at nanometer scale, which are very small. So human eye can see up to 0.1 millimeter. They can make like 2 points as distinct. The bacterium is at a distance of 1 to 10 micrometers, and meanwhile the atoms are at the distance of 0.1 nanometer, which we cannot observe. So thank you very much, class. This is it for this topic. Thank you very much for watching this video. See you in the next one.